Okay, so today we're going to talk through action potentials. Now, last lesson we looked at the resting potential. That's when the neuron is at rest. Today we're going to talk about an action potential, which is basically an impulse in a neuron. So if we have a look at the graph in front of us, we can see that when a neuron is at rest, we're looking down here, it's negative inside compared to outside. So it's minus 70 millivolts inside the axoplasm. A stimuli will cause the depolarization of that axon in the neuron. So that's when sodium ions move into the neuron to the point that they take their positive charge with them and you reach plus 30 millivolts inside the neuron. So because so many sodium ions are moving in, we hit an action potential point. So this is the point at which an impulse will be propagated so it can move along an axon at plus 30 millivolts. And then if you look at the other side, this is what has to happen in that section of the axon after that's occurred, is the repolarization. This is when potassium ions are going to move out, taking their positive charge with them, making the inside more negative. We get this small blip here of hyperpolarization, and then we go back to the resting potential of minus 70 millivolts, and at that point, we're ready for another impulse to be sent. Now, impulses always have a gap between them. Okay, if they were constant with no space between them, then our brain wouldn't be able to interpret any information from them. Okay, but every time there's an impulse, there'll be a gap between that and the next impulse. So let's have a look at how we get to the action potential. So we need some sort of stimulation of the axon to occur. Okay, so neurons are often stimulated by receptor cells. So this would be a sensory neuron. The receptor cell would cause some sodium ion channel proteins to open, and that means sodium ions would start to move into the axon and taking their positive charge with them. For example, on your tongue you have taste receptor cells, and when a particular chemical from food binds to the receptor cell, sodium ions will move in. So this first step is called depolarization. So when stimulated, the membrane potential is briefly depolarized, okay? Remember, in resting potential, it's minus 70 millivolts inside the axon, and it's more positive on the outside because all the sodium ions have built up, and they're staying there because the membrane acts as a barrier, doesn't it? So stimulation causes some sodium ion voltage gated channel proteins to open, and some sodium ions would diffuse into the axon. Now those sodium ions obviously take their positive charge with them, and they start to move the inside of the axon away from this minus 70 millivolts towards a less negative potential of 0 millivolts. Okay, but at this point, not all of the sodium ion channels are open. Now that's really important. So stimulation would cause some sodium ion voltage gated channels to open, okay, but not all. So only some sodium ions have been able to diffuse in. If the stimulus is strong enough, okay, then enough sodium ions would diffuse into the axon to reach what we call the threshold level or threshold limit. Now, threshold means enough sodium ions have moved in to move the potential difference to minus 70 millivolts inside the axoplasm. So remember, resting potential is minus 70. Sodium ions have gone and taken that positive charge with them, making it less negative. And if it gets to minus 50 millivolts, then that means we have reached what's called threshold. And the reason it's threshold is basically if you hit minus 50 millivolts, more sodium ion voltage gated channel proteins open, and therefore more sodium ions will move in by diffusion, and the cell will become more positive to reach its plus 30 millivolts, and we will have an action potential. Okay, for sure. It's basically an all or nothing response. If threshold is not reached, so if only a few sodium ion voltage gated channel proteins initially open and you only get to, let's say, minus 60 millivolts, then you will not get an action potential because you haven't reached threshold, so more sodium ion voltage gated channel proteins didn't open. Okay, at which point then your sodium potassium pump would just return the ions to their appropriate place to get them back to resting potential again. Okay, so we say it is an all or nothing response. Okay, that means either you reach threshold and you have an action potential, or you don't reach threshold, so you don't have an action potential. Also, all action potentials are a constant magnitude, so that means they're always going to be plus 30 millivolts. We're not going to get higher than this, okay? So it's not like the stronger the stimulus, the higher the action potential. What would happen is with a strong stimulus is that you'd have an increased frequency, so you wouldn't just have one action potential per second, you might have 60. Okay, which tells your brain, right, there's a much stronger stimulus that's happening here. Okay, so once you've reached your plus 30 millivolts, then that section of the axon has to be repolarized, otherwise another impulse cannot be sent. Okay, so once you hit plus 30 millivolts, okay, the sodium ion voltage gated channel proteins close at plus 30 millivolts. So now sodium ions cannot move. Remember, the membrane is 100 times more permeable to potassium than sodium ions, so sodium ions are basically stuck now inside the axon. At plus 30 millivolts, the potassium ion voltage gated channels begin to open. This is the voltage gated channels. Potassium ions will move out by diffusion and moving down the electrochemical gradient, okay, which makes the inside of the cell more negative because they take the positive charge with them. Okay, you actually reach this point where you have a bit of hyperpolarization because the potassium ions rush out so quickly, you can actually reach minus 85 or minus 80 millivolts inside the axon sometimes. And then the sodium potassium pump starts to restore the resting potential, so it moves the free sodium ions out and the two potassium ions back in, and eventually the resting potential will be restored back to here. So let's quickly talk through it with the graph. We've got resting potential at minus 70 millivolts in the axon. A stimuli causes some sodium ion voltage gated channel proteins to open. Some sodium ions would move into the axon by diffusion. That will bring up the inside of the axon so it's less negative. If you reach, let's say, minus 50 millivolts around here, which would be the threshold, okay, then at this point, more sodium ion voltage gated channel proteins open, more sodium ions move in, and you will reach plus 30 millivolts. If this threshold isn't reached, then this line would be returned back to normal, back to the resting potential by the sodium potassium pump, okay? And those voltage gated channel proteins would have closed. So let's say we did reach threshold, more sodium ions rushed in by diffusion, we reached plus 30 millivolts inside the axon. At this point, sodium ion voltage gated channels close, the potassium ion voltage gated channel proteins open, potassium ions move out by diffusion down the concentration gradient and their electrical gradient, so that's their electrochemical gradient, and we have what's called repolarization. So now we're swapping the charges back to what they were before. It starts to become more negative inside the axon. Here it's hyperpolarized because they rush out very quickly, and it reaches sometimes minus 80, minus 85 millivolts. Now the potassium ion voltage gated channel proteins will close. Remember, the sodium ion voltage gating channel proteins are closed already, and now the sodium potassium pump will restore the sodium potassium ions to their correct positions, pumping 3 sodium ions out actively and 2 potassium ions in actively until we reach the resting potential of minus 70 millivolts. And it continues to do that movement of the pumping the whole time because some ions will be leaking out and in through the normal channel proteins inside the axon.